Hello, today we'll be learning one of the most important DNA sequencing techniques called pyrosequencing. It's called pyrosequencing. Let's see what it is. In the event of nucleotide incorporation in the primed DNA with complementary base pairing rule, as you know, like adenine to thymine, guanine to cytosine, it always releases a pyrophosphate molecule, which is a combination of two phosphate molecules, as you can see in this picture. The system is designed in such a manner as we can monitor each complementary nucleotide addition using the emission of a pyrophosphate molecule. For this, we use several enzymes such as ATP sulfurylase, which in the presence of APS and with the use of pyrophosphate molecule, it can make the ATP molecule. In the presence of luciferase enzyme, this ATP molecule is sensed by that luciferase and it can create light, as we see in the fireflies. That is the basic principle we use in pyrosequencing method. First, pyrophosphate molecule, which is releasing by the nucleotide addition, is combining with APS and making ATP, and the luciferase enzyme can sense the ATP, and according to the molecules of pyrophosphate, it emits light. The increasing number of pyrophosphate molecule increase the light emission. These are the basic requirements that you need to carry out pyrosequencing. First, the DNA strand that you need to sequence along with the part that is added complementary to the primer, the sequencing primer, DNA polymerase enzyme, and the DNTPs. Along with, there are three enzymes you need to know: ATP sulfurylase and luciferase with apyrase enzymes. That's not all. You need several other substrates such as APS and luciferin. There are two main kinds of pyrosequencing techniques, as solid phase and liquid phase. Today we'll see about the liquid phase pyrosequencing method. Now let's have a look at the stepwise process of pyrosequencing. Sequencing primer is first hybridized to the single-stranded DNA template that you need to sequence and mix with the enzymes DNA polymerase, ATP sulfurase, luciferase, apyrase, and the substrates APS and luciferin. The four DNTPs are added to the reaction mixture separately and sequentially. Only one nucleotide is added once. When the DNA polymerase incorporates the complementary nucleotide onto the 3' end of the prime DNA template, a pyrophosphate molecule is released in a quantity equimolar to nucleotide incorporation. ATP sulfurylase quantitatively converts the previously released pyrophosphate molecules to ATP in the presence of APS. The produced ATP drives the luciferase-mediated conversion of generating light in amounts that is proportional to the amount of ATP. The light detected by a sensor and are recorded as a peak in a pyrogram. Each light signal is proportional to the number of nucleotides incorporated. And what happened to the excess nucleotides that are not incorporated into the chain? For that, we use an enzyme called apyrase. Apyrase continuously degrades ATP and unincorporated excess DNTPs. When degradation is complete, another DNTP is added. Addition of DNTP is performed one at a time. As the process continues, a complementary DNA strand is formed and the nucleotide sequence is determined from the signal peaks in the pyrogram. As mentioned, apyrase catalyzes degradation of ATP, unincorporated and excess nucleotides to yield nucleotide monophosphates and inorganic phosphate, resulting in a steady descending of the light output to baseline in the pyro. The intensities of emitted light or peaks of pyrogram should be analyzed to get the sequence of the DNA strand you need. No peak means no addition of the nucleotide. A single peak means addition of a nucleotide. if the peak is twice the amount of a single addition peak? Yes, that means 2 additions of the same nucleotide. Get the final sequence by analyzing the pyrogram and convert it to its complementary sequence. There you have the unknown sequence of your DNA strand. I hope now you have a good understanding about the pyrosequencing and how you can analyze the pyrogram to get the unknown sequence you need.